In 2018, the journal BMC Medicine published a prospective cohort study tracking 1,688 participants to measure the physiological effects of deliberate heat exposure. The researchers monitored a population with a mean age of 63, split almost evenly between men and women, establishing a baseline for long-term health tracking. The data revealed a staggering survival advantage. Subjects sitting in a sauna between 80 and 100 degrees Celsius, 4 to 7 times a week, experienced a 50% reduction in cardiovascular mortality. This chart isolates the dose-dependent survival advantage. Compared to subjects using the sauna once a week, the high-frequency cohort had half the likelihood of dying from a cardiovascular event. This risk reduction includes all-cause mortality, showing a parallel decrease in deaths from other non-cardiovascular events.
Subjects are achieving cardiovascular risk reductions that look identical to the effects of vigorous, load-bearing exercise, all while remaining completely stationary. Deliberate heat exposure is an exogenous stressor. It forces the body to undergo systemic biological and molecular remodeling to survive the thermal load. To understand how a hot room remodels the heart, we have to map the body's internal thermometer. You have two distinct temperatures, your shell, which is your skin, and your core, which includes your viscera, spinal cord, and nervous system. The thermal signal begins at the shell. Nerve cells in your skin contain TRP channels, or transient receptor potential channels. These are specialized sensors that detect microscopic changes in heat and convert them into electrical signals. In an extreme thermal gradient, peripheral neurons detect the heat and fire an electrical signal. This sensory input travels up the arm into a neuronal cluster at the top of the spinal cord, known as the dorsal horn. From the spinal cord, the signal ascends into the brainstem's lateral parabrachial area. Its final destination is the preoptic area, or POA, located within the hypothalamus, acting as the brain's central command center for thermoregulation. Once activated, the POA executes a dual output response. It triggers autonomic heat-dumping mechanisms, like sweating, while simultaneously communicating with the amygdala to generate a behavioral urgency to escape the heat. Below conscious control, the POA commands rapid vasodilation. Your veins and capillaries physically widen to move blood closer to the skin, increasing stroke volume. This precise neuroelectrical circuit allows a simple environmental temperature gradient to hijack the nervous system and force a system-wide cardiovascular response. As stroke volume increases, heart rate elevates to between 100 and 150 beats per minute, creating a physiological mimicry of actual cardiovascular exercise. This autonomic shift cascades into the endocrine system. A 2021 study demonstrated that moderate thermal stress, just 4 12-minute exposures, significantly decreased participants' baseline cortisol output. Biological output changes when the duration of the thermal stress is radically increased, as seen in a landmark 1986 endocrine study. Researchers subjected participants to an extreme protocol, 2 total hours in an 80 degrees Celsius environment, broken into 30-minute blocks in a single day. This graph maps the acute endocrine shock. Compared to baseline, the subjects experienced a 16-fold increase in growth hormone release. But the human body is highly adaptive. If an extreme stressor is repeated too frequently, physiological habituation limits the endocrine response. When subjects repeated the exact same 2-hour protocol on day 3, that 16-fold growth hormone spike degraded by 2/3. By day 7, the effect had decayed even further. This rapid habituation stands in sharp contrast to the cardiovascular longevity data, which requires a frequency of 4 to 7 sessions per week to achieve optimal results. Biological adaptations to thermal stress operate on divergent dose-response curves. Chronic high-frequency exposure drives heart health, but infrequent shock is required to bypass habituation and trigger massive growth hormone release. At the cellular level, high heat presents an acute biological risk. It causes proteins within your cells to denature and misfold. To prevent cellular toxicity, the body deploys heat shock proteins. These emergency molecular chaperones actively rescue and refold damaged structures back into stable shapes. The deployment of these chaperone proteins is driven by an upstream genetic program controlled by a molecule called FOXO3. FOXO3 is a transcription factor involved in DNA repair pathways and the clearing of senescent or dead cells throughout the body. High-frequency heat exposure reliably upregulates FOXO3. This constant activation of DNA repair and cellular clearing explains the 50% drop in cardiovascular mortality seen in the daily sauna users. The desire to escape the heat is a neurochemical event. This psychological discomfort signals the activation of a specific internal pathway. Intense thermal stress triggers dynorphin release, binding to kappa opioid receptors in the brain to induce agitation. The brain responds with a compensatory adaptation, triggering a subsequent proliferation of mu opioid receptors on the cell surface. Mu opioid receptors are the landing pads for the body's naturally produced feel-good endorphins. By increasing the density of these receptors, the brain increases its baseline capacity for pleasure. The acute chemical dysphoria caused by dynorphin is the required biological toll. Enduring that temporary discomfort elevates baseline mood, stress resilience, and overall endorphin efficacy. These neural, endocrine, and molecular mechanisms form a unified framework. Applied physiological stress can be steered to achieve highly specific goals. High-frequency, moderate-duration exposure activates FOXO3 to initiate DNA repair and improve cardiovascular longevity. Conversely, infrequent, high-duration shock exposure is necessary to bypass habituation and trigger massive growth hormone release. The universal variable is intensity. Pushing into an uncomfortable but safe thermal threshold releases dynorphin, forcing the subsequent opioid sensitization that improves mental health. Optimal results are not achieved by random exposure. They require matching your protocol to the specific biological pathway you intend to activate. Deliberate heat exposure is the targeted exploitation of our endogenous neural circuits to elevate human performance, cellular health, and lifespan.